Hey everyone, welcome back. So in this video, we're going to talk about gene expression. So what gene expression is the production of a protein from the DNA. So within our DNA, we have sequences that are called genes. And those gene sequences are what we use to produce proteins. So what actually produces those proteins? We have RNA. RNA is ribonucleic acid. Oh, sorry. There are three types of RNA. Messenger RNA, which is what we typically talk about in gene expression. Messenger RNA carries the message of the DNA. So I'm going to write that over here. So it carries the DNA message in a different format, a format readable by the cell. Then we have transfer RNA that carries amino acids. And remember, what are amino acids? Amino acids are the monomers of protein. So when you get a lot of amino acids together, then you can produce those proteins. And then ribosomal RNA is what actually produces proteins or assembles the amino acids, okay, into a format that can fold up into a protein. So I'll just say produces proteins by allowing the assembly of amino acids, okay? And I'm just going to put AA for amino acids, okay? I just dropped my pen. Sorry. Okay. So now let's go to the next slide. So there are two steps to transcription or to gene expression: transcription and translation. Transcription is taking the message of DNA and converting it to RNA. So we're changing the information from one language to another language. DNA can't be read by the cell, but RNA can. So this occurs in the nucleus of eukaryotes. And we convert DNA Okay, that was weird again, but whatever. DNA. It makes the video a little more exciting, right? To a language RNA that then our ribosomes can actually read. And then translation takes the RNA and converts it into a protein. So RNA is kind of like the message or the blueprints, and then the ribosome produces the protein from those blueprints. So this occurs in the cytoplasm of eukaryotes. And we're converting I'm going to put RNA blueprints Oh, that does not look like sorry, INTS into a protein. And so if we're looking, so I said nucleus of eukaryotes, cytoplasm of eukaryotes. If we're looking at prokaryotic organisms, so transcription and translation occur in the cytoplasm. So remember, in prokaryotic organisms, there's no nucleus. So all of the DNA is in that region known as the nucleoid, which is found right in the cytoplasm. So everything occurs in that cytoplasm. So this is what we're looking at. We start out with DNA. The DNA is going to be found in eukaryotic organisms. So I'll just put a E-U-K in the nucleus. If it's a prokaryote, it's in the cytoplasm, right? Within our DNA, there's going to be a region that codes for some protein. So in this case, this is what's coding for a protein. So we produce, using the template strand, which is this strand, a copy of our coding strand in an RNA format. And so that's what this is. We have our messenger RNA. Messenger RNA then can be read 3 nucleotides at a time, and that's called one codon. So we read RNA in 3 nucleotides. TIDES. Okay? And so each 3 nucleotides is called one codon. And those codons then code for specific amino acids that come together, forming a polypeptide. So these are each amino acids, and they form this polypeptide, which then can fold into a functional protein. Okay? And so let's quickly go over what RNA is, and then we'll get into actual transcription. So RNA is another type of nucleic acid. It is, whereas DNA is double-stranded, RNA is single-stranded. Okay? And it contains a phosphate, it contains the nitrogenous bases, but the pentose sugar, instead of deoxyribose, it is ribose. So this is a difference right here. We have a ribose sugar. In DNA, we had deoxyribose. So there's a difference there. There are 4 nitrogenous bases in RNA, just like in DNA. The bases are the same. We still have guanine. We still have cytosine. And we still have adenine. But instead of thymine, we have uracil. And uracil is actually, sorry, there we go. Uracil takes the place of thymine, and uracil binds to the adenine. Or I'm sorry, binds to adenine, yeah, and adenine binds to the thymine, so that's perfect. There are 3 types of RNA. There's messenger RNA, there's transfer RNA, and then there's ribosomal RNA. All of these function in protein synthesis. So we need all 3 types to produce a protein. So DNA is kind of like the blueprints of our cell. It's the coder of everything. And RNA are the workers that make those proteins. So again, here is your RNA. It is a single strand of nucleic acid. This is ribose. So remember, in DNA we have deoxyribose, in RNA we have ribose. We have guanine, we have adenine, we have cytosine, but instead of thymine we have uracil. Uracil is the one major nucleotide difference that changes the RNA from the DNA. And then the sugar, and it's single-stranded. So there's a lot of differences, but the nucleotide difference is that uracil. So what does each of those RNAs do? Messenger RNA takes a message of DNA and produces it in an RNA format that can be read by our cells, and then brings it to the ribosomes. Transfer RNA picks up the proper amino acid that's being coded for and brings it to the ribosome at the proper time. So transfer RNAs carry only one type of amino acid. They can't carry other types. So a transfer RNA, and I always like to draw it kind of like this, they have an anticodon here. The anticodon is going to be what attaches to the messenger RNA down here. And then up here, there's going to be an amino acid. And that amino acid is going to be coded for in the messenger RNA. So if the messenger RNA said AUG, the transfer RNA would have U, A, C, so it can bind because they're going to be complementary, and it's going to carry an amino acid that's coded for by the AUG. So this amino acid is going to be coded for by that AUG. The ribosomal RNA is the ribosome. So this is what makes up the ribosomes. The ribosomes function in protein synthesis. Remember, these are what make proteins. So they're kind of like the workbench of protein synthesis. And so here we have DNA, and there are multiple genes on any DNA molecule. But DNA is going to code for any specific region of DNA, and this is this region right here, which is gene 3, is going to code for some protein. Okay, so there's a code for a protein here. Our cells can't read DNA, but they can read RNA. RNA can leave the nucleus. So here we have RNA that's coded for. So here we have the DNA that is, so this would be a template, right? Here is the RNA. This would be the copy of the coding strand. And then down here, these are the amino acids that are produced. Okay? So I'm going to stop here because we'll get into how we transcribe DNA in our next video. All right? This has been a 13-minute video, so it's a perfect stopping time, I think. Bye.